So in this video, I'm going to cover the Kirby-Bauer method, day 1, which is going to be our setup. And this is also known as the disc diffusion assay or an antibiotic sensitivity test. And so this is activity 7-3. So the purpose of the Kirby-Bauer method is to perform a culture and antibiotic sensitivity test of an organism. Essentially what that means is that we want to know what antibiotic would be the best choice against a particular organism. So let's say, for example, that a patient comes in and they have a UTI, a urinary tract infection, and the doctor wants to know what drug to prescribe to that patient. So what we would do is we would first start by getting a urine culture, right? So the patient would give a urine sample. Then you could take that bacteria and streak it on a plate. And then we have these disc dispensers that has a variety of different antibiotics on it. And we would put those antibiotic discs on the plate and see which of those antibiotics is specific and will inhibit bacterial growth. Because the one that the bacteria responds to would be the one that we might choose for treatment. And so this is what we call a antibiotic sensitivity test. We're basically looking at which antibiotic would be effective against a particular organism. So this experiment would be done in pairs. You and a partner would do this together. You would use a large, what's called a Mueller-Hinton agar plate. So these are very large petri dishes, and you would be given one of these organisms. And so in this experiment, there are 6 organisms that get tested. We have Staphylococcus aureus, we have Escherichia coli, Pseudomonas aeruginosa, Serratia marcescens, Enterococcus faecalis, and Bacillus megaterium. And so each pair would be given one bacteria, and you would use a swab and you would make a lawn of that bacteria. So essentially you're going to put that bacteria all over this plate. After that is accomplished, then you would dispense the antibiotic discs onto the plate and then incubate the plate upside down at 37 degrees Celsius. And so I have a video that's going to show you this technique so that you can actually see how this is done. So here is the link for this video. And so you can watch this on your own, but I will also input this into this video.
In this video lesson, we're going to learn how to do the Bauer-Kirby method of antibiotic susceptibility testing. For that method, we use a standard size agar and plate called Mueller-Hinton agar, and these are the large 150 millimeter plates that will hold 12 different antibiotic discs. So the first thing we have to do is inoculate the Mueller-Hinton agar with the organism we're testing. Now in this case, this is one of the bacteria we're using in today's lab, Enterococcus faecalis. So we're going to be using a swab for this. We're going to remove the inoculum by flaming the tube, putting a swab in. Then we're going to wring the swab out against the inside of the tube to get rid of excess liquid. Flame the tube and replace the cap. Now we're ready to streak the plate. Now we divided our plates into thirds, as you'll see in the illustration in your lab manual to help guide our streaking. And we're going to streak perpendicular to each line, going all the way to the edge of the plate and overlapping the lines, working our way down to the center. So we lift the lid, and we're going to go perpendicular to this line. Notice I'm going all the way to the edge of the plate, and I'm overlapping the lines till I hit the center of the plate. Now we're going to rotate the plate and streak perpendicular to this line. Again, all the way to the edge and all the way to the center. Use the same swab. You don't have to dip it in again. So again, all the way to the edge, overlapping the lines. Till we hit the center. And finally rotate it to the last line and streak perpendicular to that line. all the way to the edge of the plate and all the way down to the center. And now we've inoculated the plate so that the whole plate will show solid bacterial growth next time. Of course, when we're done with the swabs, they're going to go in the biohazard container. Now we're ready to put on the antibiotic discs. Now, since this is Enterococcus faecalis, a gram-positive bacterium, we're going to use the antibiotic disc dispenser that contains antibiotics, often useful against gram-positive bacteria. You see that the lid is labeled gram-positive. So, very simple to put on the antibiotic discs. We remove the disc dispenser. It also says gram-positive on the handle. And we're going to take the lid off of the plate, put the antibiotic disc dispenser down over the plate, and push down firmly on the handle. And as you see, that dispenses the 12 antibiotic discs, and it also tamps them so they won't fall off when we turn the plate upside down and incubate at 37 degrees Celsius.
So we just looked at how to set up our Kirby-Bauer test, and now we're going to go over day 2, which is going to be the readout of this test. So again, the purpose of the Kirby-Bauer method is to perform a culture and antibiotic sensitivity test on an organism. So again, we would use this method to try and determine what antibiotic would be preferred to use against a particular bacteria, because if a patient has an infection, then you're going to need to treat with an antibiotic, and we need to know which one to use. So when we do this test is what we call standardized, and this is going to use Mueller-Hinton agar. And so this Mueller-Hinton agar is standardized in several ways, meaning that in order for the results to be meaningful, we need to have consistency when doing this test. And so there are several ways that this is standardized. The first is that the pH of this agar needs to be between 7.2 and 7.4. And the reason for that is the pH of blood. If you think about how these antibiotics typically get into our system, it's going to be through the bloodstream, whether we take it orally, right? And if we take it orally, it's going to be digested and it's going to be absorbed in the intestines, and then it's going to end up in the bloodstream. Or if it's an injection into a vein, that would also end up in the bloodstream. And so in order for this experiment to be meaningful, the antibiotics have to be effective at blood pH, because that's typically going to be the pH that those drugs are going to be found in. Next, we have soft agar for diffusion. The agar has to be soft enough that the drug is able to diffuse through the agar. So it has to be soft agar. The plate must be poured at 4 millimeters in depth, and that controls for what we call lateral diffusion. And so what that means is that if we look at this disc, the drug is going to diffuse out from the disc, right? It's going to go out. It's also going to go down and then out. So if we don't pour these plates consistently, let's say we pour this plate very, very thick. Well, if we pour our plate really thick, it's going to have to travel down longer before it travels out. And so it's not going to diffuse out as much if the plate is formed thick. If the plate is too thin, conversely, it's going to make the drug diffuse out farther. So to keep that consistent, we always pour our plates at 4 millimeters in depth in terms of the agar. And so again, that's going to control for what we call lateral diffusion, how much the drugs will diffuse outward. Plates are incubated at 37 degrees Celsius. You can probably guess the reason that we incubate them at 37 degrees Celsius. Well, that's because that's human body temperature, and these drugs are going to be used in the body to try and treat an infection. And so this test is done at human body temperature. We incubate our plates for only 18 hours. And the purpose of that is to select for young organisms that are metabolically active and dividing, and therefore would be the ones that would respond to the antibiotic. So we do this with plates that are only incubated for 18 hours to, again, select for young organisms. And then the amount of drug is standardized. So what that means is that the amount of drug will control lateral diffusion, because if I put a lot of drug on this disc, it's going to diffuse out further. If the concentration is low, it's not going to move as much. So if you think about this, the concentration of drug that's going to be on that antibiotic disc should be a concentration that is physiologically relevant, meaning that it's a dose that could be safely given to a patient, right? Because let's say that I can only give a patient 10 milligrams of a particular drug. Well, I don't want to test 10,000 milligrams in this test, because if I do that, well, yeah, the bacteria might be sensitive to the antibiotic, but that's not a dose that is physiologically relevant. It's not a dose that I can actually give a patient. So we have to also control the amount of drug that's on the antibiotic disc, again, to control for lateral diffusion and to make the results very meaningful in terms of if they could be used in a patient. And so these are the different ways that this Kirby-Bauer method is standardized so that the results are meaningful. So when we do this test, you might recall that in the video demonstrating the technique, that the organism was streaked in a lawn. So what you can see here is you see all this bacterial growth. This is a Staph aureus plate. And so this is going to be streaked, and this is all a lawn. And then here are our discs. And the discs contain the antibiotic. And again, remember that the purpose is we're trying to see if the bacteria is sensitive to that drug. So the way that we would do this experiment is that we would need to measure what are called our zones of inhibition. So our ZOI is our zone of inhibition. Basically, that's measuring the clear zone around the disc. And this is going to be recorded in millimeters. So if I look at this disc, I would measure from the edge of the clear zone across to the other end. Now, when I do this, it's important that I make sure to record in millimeters. This is a centimeter. This is 2 centimeters. This is 3 centimeters. Within each centimeter are 10 millimeters. So that means that these little lines, this is 1 millimeter, 2 millimeters, 3, 4, 5, etc. So if I'm measuring this zone of inhibition and I'm measuring the diameter, meaning the entire distance across, I would measure from here. So notice that's lined up with the 0, and it goes to about here, which you could either record as 29 or 30. It's kind of right on the cusp, right? Because if this is 3, that's 3 centimeters, which is equal to 30 millimeters. So I would record this as being 30 millimeters. Now, if you ever get into a scenario where you can't measure the diameter, meaning that the clear zone is so big that you can't really measure the entire diameter, instead, you could measure the radius, meaning from the middle of the disc out to one edge, and then multiply by 2, because you would expect that it would be equal on either side. So most of the time, though, you're able to measure the diameter. You're able to find one way in which you can measure from one edge of the clear zone to another. Now, when we get our results, we have 3 levels of susceptibility. The first, we would say that the bacteria is sensitive to the antibiotic. And what that means is that a normal dose of the antibiotic would be effective. So if I look, for example, this very large clear zone here, right, would usually indicate that the bacteria is going to be sensitive to that drug. It inhibited the growth of that microbe in this area. And so that would be a drug that would be effective to use for that infection. If we say that bacteria are resistant. So we'll go down to resistant first. If we say that the bacteria is resistant to the antibiotic, what that means is no acceptable dose of the antibiotic would be effective. So in some cases, you'll see in our examples that the bacteria might grow right up next to that disc, meaning that the antibiotic has no effectiveness. It's not effective against inhibiting that bacteria's growth. And therefore, in that case, there's no dose of that antibiotic that could be used that would be safe. So that we would say that the bacteria is resistant to that drug. It doesn't respond to that drug. The drug does not inhibit their growth. Now, in between sensitive and resistant, we have what's referred to as an intermediate. Now, if we say that a drug has intermediate susceptibility, it means that a higher than normal dose of an antibiotic is required. So again, it's like in the middle. It's not sensitive, it's not resistant, it's somewhere in between, and that it's somewhat effective, but you would need to give your patient a higher than normal dose. You might need to increase the dosage itself. You might need to extend the length of the treatment, but a higher than normal dose of the antibiotic would be required. Now, under what circumstance would you give an intermediate susceptibility drug? Well, if you think about it, let's say that you're testing a patient to see what antibiotic to give to them. And let's say that you're working with a strain of bacteria that is extremely resistant, and it's resistant to most drugs. If that's the case, if you have no drug that the bacteria is sensitive to, well, then you might choose an intermediate susceptibility because it's your best choice at that point. At least there's some response to the antibiotic. So an intermediate drug would be used if there is no sensitive organism or drug that could be used. So intermediate, you would have to use a higher than normal dose. And again, you would use that if the bacteria is not sensitive to any of the drugs. If your best option might be an intermediate, well, then you know that you need to give a higher than normal dose in order for this test to be effective. So now you'll notice that the clear zones we refer to as our zones of inhibition, right? They're zones of inhibition. True or false? So I want you to think about this first, and then once you think about it, then go ahead and push play, but it would definitely be useful for you to think about it first before hearing the answer. But true or false, all bacteria found within the clear area around the disc are dead. So that's saying that in this clear zone, that all of this bacteria is dead. So pause and think about this for a minute, and then when you're ready, push play to hear the answer. And the answer is false. It does not necessarily mean that the bacteria is dead. Remember, zone of inhibition. All we can say by looking at this plate is that the bacteria's growth was inhibited. It's not a kill zone, it's a zone of inhibition. The bacterial's growth is inhibited. And so let's talk about what are some reasons why they might not be dead. There's actually a couple reasons why they might not be dead. Reason number one, bacteria may have formed endospores, right? And so remember that in response to a harsh condition, bacteria can form endospores. And so when challenged with the drug, the bacteria may have formed endospores in this region, and they may be inhibited, right? They might not be growing, but they're not in fact dead. They could still germinate if the drug was removed. So reason number one is that bacteria may have formed endospores. Reason number two could be that the drug is what we call bacteriostatic. And what that means is that the drug inhibits growth. Stasis means to stay the same. The drug inhibits growth, but does not kill them. And so there are some instances where we would want to choose a drug that is bacteriostatic, meaning one that just inhibits their growth. And a good example of this would be for gram-negative bacteria. If you recall the composition of gram-negative bacterial cell walls, you will recall that within that outer membrane is a chemical called LPS, lipopolysaccharides. And within the LPS, part of that includes something called lipid A. Lipid A is an endotoxin. If large amounts of lipid A is released into the patient's bloodstream, it may cause the patient to go into shock. And so if we want to treat a gram-negative infection, it might be a better choice to choose a drug that is bacteriostatic, because the alternative is a drug that's bactericidal. Cidal means kill. If the drug kills the bacteria and it causes the bacteria to lyse or to break open, well, that's going to release large amounts of lipid A if the infection is caused by a gram-negative, and that's going to cause the patient to go into shock. So if we know that it is a gram-negative infection, well, then choosing a drug that is bacteriostatic, where it just inhibits their growth and therefore gives the immune system time to catch up, that is going to be preferred because, again, we don't want that lipid A released into the patient's bloodstream. And so a drug being bacteriostatic is not necessarily a bad thing. Yes, it doesn't kill them, but it does inhibit their growth. And in some cases, that's actually preferred. Now, how would we test if bacteria are actually killed within the clear zone? Well, one of the ways that we could determine if bacteria are dead within the clear zone would be to take a swab or a loop and basically just touch it to this clear zone, right? And so you take your swab or your loop and you touch it to this clear zone, and then you could inoculate a new plate. And if you inoculate a new plate where the drug is not present and the bacteria now grows, well, that tells you that there was actually bacteria in this clear zone, but their growth was just inhibited. It didn't die. If I were to swab it and then put it on a new plate and nothing grows, well, then the bacteria were dead. There were no living bacteria in that clear zone. And so there are ways that we can distinguish between those possibilities, but a good thing to know is that just because you see a clear zone doesn't necessarily mean that the bacteria are dead. It simply means that the drug is successful in inhibiting bacterial growth. So that is what we're looking for. We're looking for our zones of inhibition. So when we do this test, we have this standardized chart that we look at in order to determine if the bacteria are sensitive, resistant, or intermediate to a particular drug. So for example, if I look at this drug, on the disc, it's going to say CC2. CC2 stands for clindamycin, and the 2 is the dose, 2 micrograms. So when doing this experiment, I would look at the disc and I would see what the disc is labeled. It's going to have one of these disc codes on it. And then I would measure my zone of inhibition. So for example, if I'm looking at clindamycin, if it's less than or equal to 14 millimeters in terms of my zone of inhibition, then I would say that the bacteria are resistant to that drug, right? That clear zone is very small, and therefore the bacteria are resistant. There's no effective dose that can be used. If it's between 15 and 20, well, then we would record that as intermediate susceptibility. If it is greater than or equal to 21 millimeters, meaning again that the clear zone is very large, then we would record that the bacteria is sensitive to that drug, meaning that the drug was able to inhibit bacterial growth. So that would be what we call sensitive. And so when doing this experiment, you would measure your zone of inhibition for each of these drugs. And then you would record how large that zone of inhibition is, and then record whether it would be resistant, intermediate, or sensitive based on your numbers that you get. And so again, this is why this test has to be standardized. You have to control for lateral diffusion and all of the other things that we talked about, because you are comparing against these standard zone of inhibitions. And so if you pour your plate too thick, for example, well, that's going to affect the way that the drug diffuses, and therefore your zone of inhibition might not be the same. And so this test really needs to be standardized in order for the results to be meaningful. So here are some example plates of organisms that we would typically use in this lab. So you can see we have E. coli here. And if you look at this plate, right, you'll notice that for CC2, which again was our clindamycin, notice that the bacteria grow right up next to that drug. So your zone of inhibition is basically nothing, right? So in this case, E. coli is resistant to clindamycin. And so for each of these discs, you would label or you would record your zone of inhibition. Again, you would measure from the edge of one clear zone to the other and record that in millimeters. And so again, this is just some example plates. I do have a table where I summarize the results. So using the measurements on these plates, I will give you what the results would be. But I just wanted to kind of show you what these plates look like. So on the right here, we have Serratia marcescens. Over here, we have Pseudomonas aeruginosa. And one thing that's really obvious when you look at this plate of Pseudomonas, notice that there really is only one drug that you can choose. All of these other drugs are not effective against Pseudomonas. Pseudomonas is extremely resistant to antibiotics. It's extremely resistant. There's only one choice, and that's going to be our norfloxacin. Everything else, Pseudomonas is resistant to. And Pseudomonas is just inherently very resistant. And in fact, there have been outbreaks in a hospital before. Pseudomonas can actually grow in what are called quaternary ammonium ions, or things like Lysol, for example. That bacteria is so resistant that not only does it not inhibit it, but it can actually grow within the Lysol. And so there have been outbreaks in hospitals before where this bacteria was in the disinfectant that they were trying to use, and it grew in there. And so when they thought they were cleaning surfaces in the hospital, in fact, they were just spreading bacteria all over it. And so Pseudomonas is basically inherently very, very resistant. Here are the results for Staph aureus. So you can see that Staph aureus generally doesn't have too much resistance. Now be aware that this Staph aureus is not the antibiotic resistant strains. This is just typical Staph aureus. Not all Staph aureus are the same. There are what we call strains. There are different versions of Staph aureus that have different levels of resistance. And so that's why it's so important to do these antibiotic sensitivity tests, because yes, you could say, well, if I know that the bacteria that this patient has is Staph aureus, if that's what I'm using to guide my antibiotic treatment, it might not be effective because there are different strains of Staph aureus. And that patient may have a strain that the bacteria is resistant to that antibiotic, like MRSA, for example. MRSA, methicillin resistant Staph aureus. That is a strain of Staph aureus that is resistant to methicillin-like antibiotics. And so it's not going to respond the way that normal Staph aureus would. And so that's why these culture and sensitivity tests are so important, is because they are going to test the specific bacteria that is causing the infection. And so again, like a urine sample, because the urine sample is testing the specific bacteria that are present in the urine. If we look here, we have Bacillus megaterium, we have Enterococcus faecalis. And so again, I'm going to show you in a minute what this data looks like. So here is our data for this. So if we look, for example, at clindamycin, you'll notice that E. coli is R, which means again resistant, Serratia marcescens, resistant, Pseudomonas aeruginosa, resistant, Staph aureus, sensitive, Bacillus megaterium, resistant, Enterococcus faecalis, resistant. So notice that in this case, our clindamycin was only effective against Staph aureus. So when you're looking at this, there are reasons that we chose these different bacteria. So E. coli, Serratia marcescens, Pseudomonas aeruginosa, these are all examples of gram-negative bacteria. On the flip side, if I look at Staphylococcus aureus, Bacillus megaterium, and Enterococcus faecalis, those are all going to be gram-positive. So what that allows us to do is it allows us to compare between gram-negative bacteria and gram-positive so that we can see, is a drug only effective against one type of bacteria, or is it effective against a broad range of different organisms? And so that's why these were chosen. So we have a group of gram-negative, we have a group of gram-positive. And again, if you look at Bacillus megaterium, notice that you see S's on almost everything except clindamycin. And you might think, well, Bacillus megaterium, doesn't that form endospores? And you're absolutely right. It does form endospores. So again, those zones of inhibitions don't mean that the bacteria is killed. All it means is that the drug is inhibiting their growth. And so that's why you see so many sensitives here, because the drug is effectively inhibiting their growth, but it's not necessarily killing them. And so if we compare how resistant gram-negative and gram-positive are to the different drugs, one of the things that you're going to see is that if we look at E. coli and we count the number of drugs that that E. coli is resistant to, there are 4. Serratia has 6. Pseudomonas aeruginosa has 8. So collectively, these 3 organisms are resistant to 18 different drugs. If we compare that with the gram-positives, we only have 1 resistant for Staph aureus, we only have 1 resistant for Bacillus megaterium, and we have 5 for Enterococcus faecalis. So collectively, this has 7 resistant. So we have 18 resistant here, 18 resistant there, and only 7 resistant here. So what you'll notice is that in general, gram-negative bacteria are typically more antibiotic resistant than gram-positive. And so in the next slide, I'm going to walk you through why you might see more or less resistance for certain organisms. So, but this is just to show you that in fact, there is a difference between gram-negative and gram-positive. You do not need to memorize this table. I will not ask you which drug is effective against which bacteria, but you just want to learn how to interpret this. And so again, you need to understand that you need to measure your zones of inhibition, and then you would take those numbers and you would compare them with that standardized table, and then you would record SI or R to see if bacteria are sensitive, intermediate, or resistant. So notice if you look here, you'll see that some of these are labeled IR, intermediate or resistant. It's kind of right on the border. And so on some plates, it might measure as being intermediate. On some plates, it might measure as being resistant. But again, seeing that there are other drugs that could be chosen for Enterococcus, norfloxacin could be chosen, trimethoprim, vancomycin, well then these drugs that are IR are probably not the ones to choose. And so similarly, there's one here that's just an I. Again, if we have drugs that bacteria are sensitive to, typically those are the ones that we are going to use for treatment. We're not going to use the I ones, the intermediate susceptibility, because we would need a higher than normal dose. So let's talk about what makes an organism more resistant or less resistant to various types of antimicrobial drugs. So starting with the most resistant. The most resistant are going to be what are called prions. And prions, the reason that they're the most resistant is that they're not alive. They're not living organisms. They're not made of cells. They are simply heat-resistant proteins. So they're heat-resistant proteins. And so they're not living, so we can't target cell walls. We can't target, nucleic acids. They don't have these structures. So prions are very, very resistant. They're not alive, and in fact, they're also heat-resistant proteins. And so prions are extremely resistant. An example of a prion disease would be mad cow disease. Mad cow disease is caused by prions, and people get mad cow disease when they eat undercooked beef products of cows that are infected with this prion. And if they're infected with this prion and we eat the meat that's not been cooked, like for hamburgers, for example, if it's not been cooked thoroughly, well, then patients could end up with mad cow disease. You may remember quite a while ago, there was an outbreak of mad cow disease at Jack in the Box. The meat that they were getting and the hamburgers that they were getting were infected with these prions, and patients were getting very, very sick. Prions are very, very resistant. Next on our list of most resistant would be our endospores. And remember that that's because endospores have that tough keratin shell. Remember that that keratin is a tough structural protein that's also found in skin. And so endospores have that tough keratin shell, which helps make them very resistant. And this is why bacteria produce endospores, because they're going to produce them in response to a harsh environment. And so they're resistant because of their tough keratin shell. Next, we have our mycobacteria. And our mycobacteria, remember, would be our acid-fast positive bacteria. And for mycobacteria, the reason they are resistant is because they have that waxy cell wall with 60% mycolic acid. You might recall that when we did our acid-fast stain and we did our endospore stain, that we had to steam during the addition of the dye in order to get the dye into the cells. And so both endospores and mycobacteria have these outer structures that make them very resistant. And that's why when we stain those organisms, we have to use steam while applying the dye so that the dye can penetrate those structures and get in. So endospores are very resistant. Mycobacterium is very resistant, but a little bit less than endospores. Protozoans, this is what we are going to be learning currently. Protozoans have two main stages, what we call a cyst stage and what we call a troph stage, or vegetative protozoans. The cyst is going to be the most resistant. And the cyst is going to be the most resistant because it has a thick wall on the outside that is made of chitin. You might recall that chitin is that tough polysaccharide. It's found in the exoskeleton of arthropods, for example. But that chitin is it's a structural protein. And so as a result, the cysts are going to be very resistant. You'll also see this when you learn about protozoans, that any of the ones that are transmitted by fecal-oral route, meaning that you ingest fecally contaminated food or water, the stage that you ingest that's going to cause the disease is going to be the cyst, because the cyst is the one that's resistant and can withstand stomach acid. If we look at the vegetative protozoans, they have a pellicle, which is like a cell wall, but it's not in fact a cell wall. And so the vegetative protozoans are going to be a little less resistant than the cyst. The cyst is the one that's more resistant. If we look at gram-negative bacteria are going to be fairly resistant because they have an outer membrane with porins, and those porins regulate what goes in and out. Additionally, gram-negative bacteria are more likely to be bacteria that produce capsules or slime. And so gram-negative are going to be more resistant than gram-positive. And you might recall that in the table that we just looked at, we saw that gram-negative was much more resistant than gram-positive. And so it's because the gram-negative bacteria have that outer membrane with those porins in it. For our fungi, including fungal spores, those have some resistance because, again, they have a cell wall made of chitin. It's a tough structural protein. When we look at viruses are either non-enveloped or enveloped viruses, and viruses without a lipid envelope are going to be more resistant because there's no envelope to be dissolved by a lipid-soluble antimicrobial, meaning that the way that some of these drugs work is that, or these chemicals work, is that they dissolve that outer membrane. And if they dissolve that envelope, well, that's how the virus is going to get into the cells. So if we can dissolve those envelopes, well, then the bacteria is not going to be the virus is not going to be very resistant. So not resistant would be gram-positive and viruses with lipid envelopes, because if they have a lipid envelope, they can be dissolved. The lipid envelope could be dissolved by the chemical, and therefore the virus is inactive. It's no longer able to infect a host cell. Viruses without that envelope, they're much more resistant because we can't simply dissolve their envelope. They don't have one. And so this is basically just showing you from most resistant down to least resistant. And you're seeing this in other parts of the course as well. So you'll also hear this again. So if we look at our antibiotics, we can kind of break down antibiotics into two main categories. We can call a drug a broad spectrum drug. And that means that it kills or inhibits both gram-positive and gram-negative. Broad spectrum means it targets a variety of different microbes. It's not only targeting one type, it's targeting multiple types of microbes. There are pros and cons between using a broad spectrum or a narrow spectrum drug. A broad spectrum drug might be preferred if, for example, your patient is extremely sick and needs treatment right away, meaning we don't have time to figure out what antibiotic would be effective. If that's the case, if we're in a life or death situation and the patient needs the drug right away to get rid of that infection, the best approach might be to give the patient a broad spectrum because it's going to target a wide variety of microbes. So even though we don't know what's causing the infection, at least that drug hopefully should inhibit the bacterial's growth. Now, while it's useful in some cases, again, if you need a drug right away, it's not always the preferred method. It's not always the preferred choice. And the reason is that we don't want to overuse broad spectrum because it kills normal flora. So it kills bacteria that's part of your body normally. And so we don't want to get rid of that normal flora because that normal flora in many cases are actually beneficial bacteria. They are bacteria that are doing something useful. And so one of the side effects that you'll see sometimes with a broad spectrum is that for females, for example, you might end up with a yeast infection as a result. And that's because when you take that broad spectrum, you're not only getting rid of the bad bacteria, but you're getting rid of the good bacteria too. And when you do that, now you're getting rid of the good bacteria that are normally competing with other organisms for limited resources. So in the case of a yeast infection, when a female takes a broad spectrum drug, she loses a lot of bacteria that's part of her normal flora in the vagina. And as a result, the yeast that's always there, but is kept in check by the bacteria, when that bacteria is now gone, the yeast has a little party and says, woohoo, food, space, and the yeast starts to grow, and that leads to a yeast infection. And so, broad spectrums have a use, but they also have a drawback. They're not always preferred because they're not only getting rid of the bad bacteria, but they're getting rid of the good bacteria too. And this can also lead to what we call a superinfection. And a superinfection is an infection following a previous infection, usually by microorganisms that have become resistant to the antibiotic used earlier. So basically, it's a secondary infection. And so if we're using a broad spectrum and it's not killing all the bacteria, let's say that you don't finish the drug, for example, well, you're getting rid of the weaker bacteria first, and the ones that are more resistant are more likely to survive and then grow, and now you end up with a population that is highly resistant. And they are resistant to that drug that was used previously. And so again, a broad spectrum is not always the preferred choice. Yes, it's going to kill, a variety of different microbes, but in some cases, that's not always a good thing. We don't want to get rid of all bacteria in the body. We need those bacteria. A narrow spectrum drug is one that kills or inhibits only specific types of bacteria, meaning they might only target gram-positive bacteria, for example. And so an example of this would be penicillin. Penicillin is only effective against gram-positive because it targets peptidoglycan. And remember that gram-positive have a thick layer of peptidoglycan. Gram-negative, on the other hand, has a much thinner layer of peptidoglycan. So for narrow spectrums, they only target certain types of bacteria. And so again, that can be a good thing because it's not going to disrupt normal flora. It's more targeted towards the bacteria that is causing the infection. And oftentimes, as a result of it being more targeted, it's also more effective because it's only inhibiting bacteria that are causing the infection or bacteria that are likely to cause the infection. And so oftentimes patients will get better faster with a narrow spectrum drug because it is more targeted. So again, there are times where a broad spectrum would be preferred. There are times when a narrow spectrum is preferred. It really depends on the situation. If we have time to figure out what's causing the infection, well, then choosing a narrow spectrum drug may be the way to go because then it's targeting that specific bacteria. If, on the other hand, the patient is very, very ill and they need a drug right away, then a broad spectrum might be the way to go. And so these are different types. So let's look at our data and see which of the drugs that were tested were broad spectrum and which ones were narrow spectrum. So if we look at our data, if we look at clindamycin, for example, notice that the only bacteria that is sensitive to clindamycin is Staphylococcus aureus. This is extremely narrow spectrum, very, very narrow spectrum. Notice it's not effective against all gram-positives. It is specific to Staph aureus. Clindamycin is typically used in topical acne treatments. So it's a drug that is used on skin for people who have acne. Staph aureus is a skin type bacteria. So notice that it is sensitive, Staph aureus is sensitive to clindamycin. So clindamycin would be a narrow spectrum. If I look at penicillin, notice that penicillin is effective except, or bacteria that are sensitive to penicillin would be Bacillus megaterium and Enterococcus faecalis, both of which are gram-positive. So again, penicillin would be a narrow spectrum because it's only targeting gram-positives. It has no effect on gram-negatives. And then lastly, we have our vancomycin. Vancomycin, notice, only targets gram-positive, but does not target gram-negative. And so this would be another example of a drug that is a narrow spectrum. So the yellow boxes indicate drugs that are narrow spectrum. Let's look at which drugs are now broad spectrum. So one that really jumps out is norfloxacin. This is in the quinolone family of antibiotics, which you'll learn about later. And what you'll see is that basically it was effective against all 6 bacteria that we tested. E. coli was sensitive to it, Serratia was sensitive to it, Pseudomonas was sensitive to it. Notice that's the only drug that Pseudomonas was sensitive to. So, norfloxacin is very, very broad spectrum. It can be used against a wide variety of different bacteria. That is a good and a bad, right? So again, it can be a downside because it gets rid of normal flora. And we don't want to overuse drugs that are very broad spectrum because they're useful, right? If you have a patient that has a Pseudomonas aeruginosa infection, well, you better hope that it's sensitive to that norfloxacin. It's the only drug to choose. If we overuse norfloxacin and bacteria become resistant to it, well, then we're going to be in trouble for Pseudomonas infections because we have now lost our only drug that Pseudomonas is sensitive to in this case. And so norfloxacin should only be reserved for cases where it's absolutely necessary. Again, because we don't want to get more antibiotic resistance to those broad spectrums. They have a use, but they shouldn't be overused. Another example of a fairly broad spectrum would be trimethoprim, which is labeled on the disc TMP5. Notice that the only organism that was resistant to the trimethoprim was the Pseudomonas aeruginosa. In every other case, bacteria were sensitive to the trimethoprim. So that would be another example of a broad spectrum. Now, again, you're not going to have to memorize this list and tell me which ones were broad and which ones were narrow. That I don't care if you memorize that detail. But if given plates and they're labeled with R's and S's, you should be able to determine whether or not it's a broad spectrum drug or a narrow spectrum. And the way that you do that is that you look to see for any given disc, for example, let's say it was CC2, you would look at that CC2 disc and see if it says R or S. If it has, a bunch of S's, if it's sensitive for both gram-positive and gram-negative, well, then you're looking at a broad spectrum. If only one bacteria responds or only one type, meaning only gram-positives were sensitive to it, well, then it's a narrow spectrum. So again, you don't have to memorize this list, but if given pictures of plates, you should be able to say whether or not that drug is going to be broad spectrum or narrow spectrum. And so when we talk about broad spectrum or narrow spectrum, we're referring to the drug itself. Is the drug targeting a variety of bacteria or only one type? When we talk about sensitive or resistant, we are talking about the bacteria, the organism. Is the organism sensitive to that drug? It's not, is that drug sensitive to the E. coli? Is E. coli sensitive to the cephalethin, for example? So just be aware that when we're doing the antibiotic sensitivity test, we're testing if bacteria are sensitive to those drugs. And so that's what we're looking at. Now, another phenomenon that we see when looking at a Kirby-Bauer method is we can see what's called drug synergy. And 2 drugs display greater effectiveness together than either drug alone. For example, if one drug has an effectiveness of 4 out of 10 and the other 3 out of 10, right? Then you would expect that combined it would be 7 out of 10. Yet when you look at it, it's actually 10 out of 10. They're more effective when combined than either drug individually. And so an example that you can see here is here's your disc, here's your clear zone, here's your clear zone. And notice that if I were to look across this disc, my zone of inhibition should end roughly where this dotted line is. Yet we still see a zone of inhibition in between there. This is an example of drug synergy. This drug synergy is because when these two drugs are combined, it's more effective than either drug alone. And so this would be drug synergy. So if we were to look at this on our plates that we did, if you were to look at this plate, which two drugs on this plate do you notice exhibit drug synergy? And so if we look at trimethoprim, this red dotted line, so notice it's going across, you would expect that this zone of inhibition ends here. And if we look at this disc that's labeled G25, based on the fact that it's this much on this side, you would expect it's the same on this side. So my zone of inhibition should only be here. Yet look at all this additional clearing that we have in between. So we would say that these two drugs exhibit drug synergy. Combined, they're more effective than either drug alone. And so the drugs that are on this plate would be this trimethoprim, which is labeled TMP5, and sulfoxazole, which is labeled G25. These two drugs, when combined, are more effective than either drug alone. And in fact, when these are combined, they make up something called Bactrim, and Bactrim is used for urinary tract infections a lot. And when combined, when these two drugs are combined, only about 10% of the normal dose needs to be used when you combine them. So it allows you to give a lot less of the drug than if you were to give them individually. And so they're combined in Bactrim because these two drugs display synergy. Combined, they're more effective than either drug alone. And so let's look at why. So if we look at this, we have here PABA, para-amino benzoic acid. This is an essential nutrient used by many bacteria to make folic acid. And so this PABA, normally PABA gets converted into this dihydrofolic acid. The dihydrofolic acid gets converted to tetrahydrofolic acid. So basically through this process, bacteria are synthesizing their folic acid. Now, why do they need folic acid? Well, folic acid is used to make nucleic acids. It's used to make DNA and to make RNA. For humans, we don't synthesize folic acid. We get our folic acid from our diet. We need folic acid too to make our nucleic acids. But bacteria, on the other hand, they use PABA, and they will convert PABA into folic acid. Now, the way that the sulfa drugs work is that they inhibit this step going from PABA to dihydrofolic acid. In the metabolism chapter, you'll learn that the sulfa drugs are competitive inhibitors for the enzyme that converts PABA to dihydrofolic acid. And so this sulfa drug is going to inhibit dihydrofolic acid from being produced. So if we don't produce the dihydrofolic acid, then we can't produce the tetrahydrofolic acid. The reason that the sulfa drugs and trimethoprim are synergistic is because trimethoprim inhibits a different step in the process. It inhibits the step going from dihydrofolic acid to tetrahydrofolic acid. And so the sulfa drug inhibits this step, trimethoprim inhibits this step. So when combined, they're much more effective because it's hitting this pathway in multiple places and ensuring that folic acid can't be synthesized. And if bacteria can't synthesize that folic acid, then they can't grow because they can't make their nucleic acids. And so this is why these two drugs display drug synergy. They display drug synergy because they target two different steps in the same pathway. They are basically ensuring that folic acid doesn't get produced. And if folic acid doesn't get produced, well, then if the bacteria can't make folic acid, they can't make their nucleic acids and they can't grow. And so that's why these drugs are antibiotics, because they're inhibiting bacterial growth by targeting, in this case, this metabolic pathway. And so these two drugs display synergy. They are more effective combined than either drug alone. And so again, those drugs go under the trade name of Bactrim. And again, only about 10% of the drug is needed when you combine them. And so this would be an advantage. And so this concludes our Kirby-Bauer method.